September 22, 2017. Future Science Group FSG today announced the publication of an article in Future Science OA presenting early data from a novel assay for the non-invasive detection of pd-l1 and other biomarkers in patient blood samples. Response rates to immunotherapies targeting the PD-1 pathway vary, and efforts are ongoing to improve the discovery of those who will and will not benefit from such therapies. Expression of pd-l1, among other biomarkers, is associated with response. However, owing to tumor heterogeneity and the fact this biomarker is not static, biopsies are not suitable. Furthermore, biopsies are invasive and unsuitable for repeated testing. Novel research from an international team led by Jinkai Teo Merck Research Laboratories, Singapore sought to solve this problem using peripheral blood samples and a less invasive approach. Whole blood from both healthy donors and breast cancer patients underwent circulating tumor cell enrichment and was loaded onto a microfluidic chip, undergoing chip cytometry. The results demonstrated that the workflow had a mean detection rate of 22.8% and could determine pd-l1 and pd-l2 expression levels. We believe the main advantages of chip cytometry lie in the iterative staining process that allows retrospective evaluation of additional markers and the potential to measure a large number of parameters without the spillover compensation problems encountered with flow cytometry, commented the authors. This approach allows the analysis of additional immunomodulatory targets on tumor cells beyond pd-l1 and pd-l2, which is particularly critical, considering high-dimensional analysis of these markers is likely to become increasingly relevant as immunotherapy moves beyond the administration of single immunomodulatory agents toward combinations that synergize in their anti-tumor immune response. Furthermore, the potential to include more positive or negative markers could allow increased confidence that identified cells are CTCs. However, the authors note that these data are preliminary and further experimentation is needed to fully establish feasibility of the approach. And available from https www.futurescience.com. 10.4155 FSO 820,170,079 About Future Science OA launched in March 2015. Future Science OA is the inaugural gold open access journal from Future Science Group. It publishes articles covering research of application to human health and utilizes a CC/BY license. Future Science OA embraces the importance of publishing all good quality research with the potential to further the progress of medical science. Both negative and irreligious research is considered. The journal also features review articles, editorials and perspectives, providing readers with a leading source of commentary and analysis. More open access oncology research can be found at https www.futurescience.com. Journals category Oncology. About Future Science Group. Founded in 2001, Future Science Group FSG is a progressive publisher focused on breakthrough medical, biotechnological, and scientific research. FSG's portfolio includes 2 imprints, Future Science and Future Medicine. Both publish ebooks and journals. In addition to this core publishing business, FSG develops specialist e-communities. Key titles and sites include Bioanalysis Zone, Epigenomics, Nanomedicine and the award-winning Regenerative Medicine. The aim of FSG is to service the advancement of clinical practice and drug research by enhancing the efficiency of communications among clinicians, researchers and decision-makers, and by providing innovative solutions to their information needs. This is achieved through a customer-centric approach, use of new technologies, products that deliver valuiformity and uncompromisingly high standards. http www.futuresciencegroup.com. Future Science Group. Related tumor cells article. Tumor-dwelling immune cells thwart cancer immunotherapy. Researchers have caught tumor-associated immune cells called macrophages in the act of stealing checkpoint inhibitor antibodies away from their intended T-cell targets, and blocking this thievery led to improved therapeutic responses in tumor-bearing mice. Are tumor cells glutamine addicts? Many tumors are thought to depend on glutamine, suggesting glutamine deprivation as therapeutic approach, but a new study shows that this effect might have been overestimated. Tumor cells are dependent on fat to start metastasis. A study headed by Salvador Esner Benita, a researcher at the Institute for Research in Biomedicine, Barcelona, and published today in Nature, identifies metastasis-initiating cells through a specific marker, namely the protein CD36. Current tumor cells news and events.